So now we are going to discuss about the structural and functional unit of the kidney, which is known as nephron. So nephron is a structural and functional unit of the kidney. Okay, each kidney is made up of approximately 1 to 1.2 millions of nephron, and the length of the nephron is approximately 4 to 6 centimeter, and the diameter is approximately 200 micrometer. Okay, so if you see that, this is called as the first component of the nephron. So this is known as the first part called as multigan body. Can you see this portion? This is known as multigan body. So each nephron is going to show the presence of multigan body, first part. Then there is a narrow neck. Okay. Then there is a part which is coiled, which is known as proximal convoluted tubule. Okay. After the proximal convoluted tubule, you can see there is a next part which is known as the Henle's loop or loop of Henle's. Okay. After loop of Henle's, the next part, which is again a coiled. So the distal part which is coiled is known as distal convoluted tubule. And finally, there is a collecting tubule. So these all are parts of nephron. So nephron plus collecting duct. Can you see this is a collecting duct? So collecting duct as well as nephron, it is together known as uriniferous tubule. Why it is called as uriniferous tubule? Because it is concerned with the formation of urine. Okay, it is concerned with the formation of urine. So very first part you can see over here, this is known as the multigan body. Okay, another name for the multigan body is, it is also known as the renal corpuscle. So either you can call it as multigan body, or you can also call it as renal corpuscle, or it is also called as pygmalion corpuscles. It is one and the same. Okay. Now, in case of the multigan body, it is further made up of two components. One, you can see there is a cup-like structure. This cup-like structure is called as Bowmans capsule. This cup-like structure, okay, which is a blind-ended, it is formed by the invagination. Okay, invagination, that means suppose if the tube like this, you can see over here, if this is a tube like this. Okay, now if this part will go inside, so you can see the structure will become like this, right? So this process is known as invagination. Okay, so imagine there is a balloon, and if you put a finger over here, so this part will goes inside. So this process is known as invagination. So you can see there is a structure called as Bowman's capsule. So the Bowman capsule is formed by a process called as invagination. So this is a cup-like structure. Okay, this cup-like structure encloses a network of capillaries. Okay, now that network of capillaries or bundle of capillaries or bunch of capillaries known as glomerulus. So there can be 50 capillaries. So network of capillaries plus a cup-like structure, that is Bowman's capsule. So network of capillary is known as glomerulus. Cup-like structure is called as Bowman's capsule. So these two structure together, they are going to form multigan body or renal corpuscle or pygmalion corpuscle. That is the first part. Okay. Now glomerulus is formed by the renal artery. Renal artery is going to enter inside the kidney. The renal artery is going to show the many branches. This is called as afferent arteriole. Okay, it should be afferent arteriole. That afferent arteriole is going to enter inside the Bowman capsule. It is going to divide, redivide, and it is going to form network. Again, it is going to join, rejoin. So that network is called as glomerulus. Again, it is going to join, rejoin, and it is going to form afferent arteriole. So this is called as afferent. Afferent is one which is going to enter inside. This is known as efferent. Efferent is one which is going to leave. Okay. One which is going to enter is called as afferent, and one which is going to leave is known as efferent. So this is known as a bundle of capillaries, okay, which is called as glomerulus. And then this is called as cup-like structure. That cup-like structure is called as Bowman capsule. If you see that cup-like structure, it has got two layers. You can see there is inner layer which is very close to the glomerulus is known as visceral layer. Okay, you can see there is the outer layer. That outer layer is this layer, which is called as parietal layer. So Bowman capsule has further characterized by the presence of two layers. Okay, so that is called as inner layer and outer layer. So this is a complex structure which is having the double wall. Inner wall is made up of squamous epithelium. Okay, outer wall is called as parietal, which is also known as visceral layer. All right, and there is a space between outer and inner. There is a space. Can you see? There is a space. That space is known as capsular space or urinary space. Okay, again I am going to repeat. Bowman capsule is a complex structure. All right, it is going to show the outer layer and inner layer. Outer layer is called as parietal layer. All right, it is made up of squamous epithelium. Okay, inner layer is known as visceral layer. Okay, this visceral layer is made up of specialized epithelium. That epithelium is called as podocyte. Okay, why podocyte? Because they are having a foot-like projection, a finger-like projection. Okay, so these foot-like projection, they are in which contact with the glomerulus. Again, outer layer is called as parietal layer, inner layer is called as visceral layer. Both layer are lined by the squamous epithelium. Okay, the inner layer is called as visceral layer. Visceral layer is going to show the presence of a specialized cells called as podocyte. Okay, which are modified squamous epithelium, which is going to show the finger-like projection. Between the outer layer and inner layer, there is a space. You can see this is a space. That space is known as urinary space or capsular space. Okay, that space is known as urinary space or capsular space. Okay, so the first filtration of the blood will take place in multigan body. The first filtration of the blood that will take place in the multigan body. Okay, so this is all about the structure called as multigan body or renal corpuscle or Pygmalion corpuscle. Coming to the next part, this part is called as neck. Okay, can you see there is a narrow neck? Now that neck is lined by ciliated epithelium. Okay, that neck is lined by ciliated epithelium, and whatever the lumen of the neck, lumen is the space in the neck, is known as urinary pole. Okay, this is known as urinary pole. What is the second part? Second part is called as neck. Neck is lined by ciliated epithelial cells. Okay, whatever the cavity or whatever space that is present within the neck is known as urinary pole. Okay, it is known as urinary pole, and that neck is going to open into the anterior portion, which is a coiled portion, is known as PCT. PCT stand for proximal convoluted tubule. Okay, so neck is going to open into the proximal. Proximal is anterior, convoluted, meaning of the convoluted is coiled, and smaller version of the tube, so it is called as tubule. Okay, so neck is going to open into the proximal convoluted tubule. This is highly coiled part. It is lined by the cuboidal epithelial cells with microvilli. Since the microvilli are present, these microvilli are concerned with the reabsorption. That means some important substances are reabsorbed in proximal convoluted tubule. Okay, so the neck is going to open into the proximal convoluted tubule. Proximal convoluted tubule internally lined by cuboidal epithelial cells, which is going to show the presence of brush border or microvilli. This proximal convoluted tubule is concerned with the selective reabsorption. Some important substances are absorbed inside the proximal convoluted tube. Because of the coiling, the filtrate flows slowly and it is going to spend more time in proximal convoluted tubule so that it will ensure the maximum absorption. Okay, why this is coiling? Because the fluid will spend more time in the PCT, and that is going to ensure the maximum reabsorption of the substance. Now that proximal convoluted tubule is going to open into the U-shaped tube known as Henle's loop. Now the Henle's loop is going to be represented by the descending limb. Can you see this is a descending limb which is a thin walled. This is the ascending limb which is a thick walled, and both are joined by the U loop. So this entire structure is known as Henle's loop. This entire structure is known as Henle's loop. Okay, so you can see this is a descending limb which is thin walled, and this is permeable to water. Okay, this is thick walled. It is not permeable to water, but it is permeable to salt. Are you getting this? So these both, that is descending as well as ascending, they are again lined by the squamous epithelial cells. Okay, ascending limb is thick walled and impermeable to water. Okay, now this is lined by the squamous epithelial cells. Okay, whereas this is lined by the cuboidal epithelial cells. Okay, now you can see over here, this is called as Henle's loop, and that Henle's loop is surrounded by a blood capillary. Can you see there is a blood capillary? So the blood capillaries which is going to surround the Henle's loop is known as Vasa recta. Okay, the capillaries which are going to surround Henle's loop is known as Vasa recta. So blood is going to flow in one direction, whereas the urine is going to flow in opposite direction in case of Henle's loop. So now it is known as counter current mechanism, where both the fluid are going to move in opposite direction. Okay, now Henle's loop. So what is a Henle's loop? Henle's loop is U-shaped tube. All right, it is made up of descending and ascending limb. Descending limb is thin walled, ascending limb is thick walled. Okay, the descending limb, it is permeable to water. It is lined by the squamous epithelial cells. Then ascending limb, it is not permeable to water. It is lined by the simple cuboidal epithelial cells. Now Henle's loop is surrounded by the blood capillaries known as Vasa recta. Finally, that Henle's loop is going to open into the next coil tube. So the posterior portion of the coil tube is known as, that is, distal convoluted tubule. So distal is posterior, convoluted is coiled, and tubule, smaller version of the tube. Now distal convoluted tubule is also lined by the cuboidal epithelial cells, but there are no microvilli. So it is made up of only simple cuboidal epithelium. It is made up of only simple cuboidal epithelium. Now DCT is concerned with the tubular secretion, active secretion, right, as from the surrounding capillaries, and it is also going to help in absorption of water and regulation of the pH of the body. Finally, that DCT is going to open into the small tube, small short tube known as collecting tubule. Can you see this is a collecting tubule? Okay, so this collecting tubule are going to open into the collecting duct. This collecting duct is going to receive many collecting tubule. So collecting duct is a common duct where many nephrons are going to open. Okay, so such 7 to 8 collecting duct, they will come together and then they are going to form duct of Bellini. Okay, such 7 to 8 collecting duct, they will come together and then they are going to form duct of Bellini. Okay, so this is called as structure of nephron. So as far as the position of the nephron is concerned, you can see this is how the nephron is located inside the kidney. You can observe that there is a multigan tubule. This is called as proximal convoluted tubule. This is known as distal convoluted tubule. So multigan tubule, proximal convoluted tubule, and distal convoluted tubule, all these parts are located in the cortex. Can you see over here? Whereas Henle's loop or loop of Henle and collecting duct are going to, or they are present in the medulla. Multigan body, TCT, DCT are located in the cortex of the kidney, whereas Henle's loop collecting duct or collecting tubules are going to open or they are present in case of medulla. So ultimately that urine is collected into the collecting tubule to the collecting duct. The collecting duct is going to open into the duct of Bellini. 7 to 8 collecting duct is going to open into the duct of Bellini. This duct of Bellini is going to open over here. That is a renal papilla. A renal papilla is going to open into the minor calyx. Minor calyx is going to open into the major calyx. Major calyx is going to open into the pelvis and finally into the ureter. Okay. Now there are two types of nephron. Okay, what are the types of nephron? One is called as cortical nephron, another is called as juxtamedullary nephron. So what do you mean by cortical nephron? It is present in the cortex. So you can observe that it has got shorter loop of Henle. Okay, that is the first thing. Then it is mostly located in the cortex and it is going to form 80% of the total nephron. Another nephron is called as juxtamedullary nephron, where it has got longer loop of Henle's. So it is located in both cortex as well as medulla. All right. The loop of Henle is longer and it is going to form approximately 20% of the total nephron. Since it has got longer loop of Henle, so obviously Vasa recta is also present. So there are very few chances that Vasa recta is present in case of cortical nephron. Sometimes what happens that DCT, okay, sometimes what happens that DCT stand for distal convoluted tubule. So sometimes smooth wall, okay, sometimes this is called as a glomerulus, okay, this is called as afferent arteriole. So sometimes the smooth muscle cells of the afferent arteriole, this is the smooth muscle cells of the afferent arteriole, they are modified in a such a way that their cytoplasm or their sarcoplasm become granular, and these cells are now known as juxtaglomerular cells. Okay, क्या बोलने का? It is called as juxtaglomerular cells. So sometimes the cells of afferent arteriole, that is muscle cells of the afferent arteriole, so their cytoplasm or their that is sarcoplasm, that sarcoplasm become granular and now it is known as juxtaglomerular cells. Okay, so in each nephron, beginning part of the DCT makes the contact. So this is, you can see, this is called as distal convoluted tubule. Can you see this is a distal convoluted tubule? So this distal convoluted tubule make the contact with the afferent arteriole at the same position. Okay, or at the same nephron. So cells are the cells in the wall of the DCT are densely packed. Can you see it is a densely packed over here? So these densely packed cells are called as macula densa. Cells of the DCT near the juxtaglomerular cells, they are densely packed. Now it is known as macula densa. Okay, now this is known as macula densa. So macula densa and juxtaglomerular cells, they are going to form the apparatus called as juxtaglomerular apparatus. Okay, that is going to form a juxtaglomerular apparatus. Again, I am going to repeat, pay attention. The afferent arteriole, it has got smooth muscle cell in the wall. These smooth muscle cells are modified, their sarcoplasm become granular. Now these cells are known as juxtaglomerular cells. Sometimes that DCT make the contact with the glomerulus. Okay, so that DCT comes in contact with the afferent arteriole of same nephron, right? And the cells of the DCT, you can see these cells of the DCT, they are densely packed over here. So these densely packed cells are called as macula densa. Okay, the densely packed cells are called as macula densa. So macula densa and juxtaglomerular cells, these together, they are going to form a structure known as juxtaglomerular apparatus, which is going to secrete some important hormone as well as enzyme. Okay, so this is all about juxtaglomerular cells. Okay, so juxtaglomerular apparatus is made up of juxtaglomerular cells of the afferent arteriole and macula densa cells of distal convoluted tubule. Okay, easy. So this is all about the structure of nephron. Hmm? So now we are going to discuss about blood supply to the kidney. Okay, as we know that kidney is going to receive the renal artery. You can make out in the diagram. Can you see there is a renal artery? So this renal artery is a branch of dorsal aorta. Can you see the dorsal aorta? I am going to show over here. Okay, this is a dorsal aorta. Dorsal aorta has a branch called as renal artery. Renal artery is going to enter in your kidney. It is going to divide to form afferent arteriole. Can you see this is a afferent arteriole? This afferent arteriole is going to enter into the Bowman capsule. It is going to divide, redivide to form a structure called as glomerulus. Okay. Again, glomerulus, it is going to come out as efferent arteriole. Can you see this is a efferent arteriole? Now that efferent arteriole, which is going to surround the renal tubule, it is known as peritubular capillaries. So this is entire a peritubular capillaries. This is the entire peritubular capillaries. Part of the peritubular capillaries, which is parallel to Henle's loop, is known as Vasa recta. Can you see the location of Vasa recta? Part of peritubular capillaries, which is parallel to Henle's loop, is known as Vasa recta. So this is a Vasa recta. Okay. Now this peritubular capillaries and Vasa recta, they are going to join renal venule. Can you see this is a renal venule? Renal venule is going to join renal vein. Can you see the location over here? This blue one is renal vein. This renal vein is going to enter into the inferior vena cava, and inferior vena cava is going to carry deoxygenated blood to the heart. Again, from the heart, oxygenated blood is carried by the dorsal aorta. Dorsal aorta has got further branches called as renal artery. Renal artery is going to enter into the kidney. After entering into the kidney, it is divided to form branches called as afferent arteriole. Afferent arteriole is going to enter into the Bowman capsule. It is going to form glomerulus. Okay, then it is going to leave the Bowman's capsule. Now it is called as efferent arteriole. Efferent arteriole is going to enter, or it is going to surround the nephron. Okay, so it is called as peritubular capillaries. Peri is surrounding. So it is going to surround the renal tubule. So peritubular capillaries. Part of the peritubular capillaries which is going to surround the Henle's loop is called as Vasa recta. Okay. Ultimately, they are going to again join to form renal venule. This renal venule is going to join a renal vein. A renal vein is going to leave the kidney, and then it is going to join the inferior vena cava. This inferior vena cava is going to carry deoxygenated blood to the heart. So this is known as blood supply to the kidney.